TTP, there is different types of color pattern, CTP, GTP. So the sequence of nucleotides is determined by the analyzing the color patterns of the fluorescent sequence. So then going for the data analysis. So in data analysis, so there is a sequence assembly. The short sequence reads are assembled into the longer contiguous sequence contigs using bioinformatic tools, and there is a variant calling. So the variance calling the assembled sequence and compared to the reference genome to identify the genetic variations. Okay. So what happens? The short sequence reads are assembled into the longer contiguous sequence, which is the contigs using. So there is one kind of bioinformatic tools are used to read the short sequence, and there is some kinds of variance calling are there. So whatever in the variance calling, so the assembled sequence are compared to the reference genome to identify the genetic variances. So there is some kinds of database, right, where there is a reference genome are already stored. So just we are compared the sequence with the reference sequence to identify the genetic variations such as the single nucleotide polymorphisms are the indel variants are there. Then we go for the data interpretation. So the identified genetic variation are analyzed to gain insight into the biological processes, the disease mechanisms, and the evolutionary relationship. So what about the advantages of NGS? So the first advantage is the high throughput, high throughput. So mainly the NGS can sequence billions of base pairs of DNA in a single run. Next is the reduced cost. The cost of sequencing has decreased significantly, making NGS to more accessible to the researchers. Increased sensitivity. So definitely NGS can detect the rare variants, okay, which cannot be detected through the Sanger sequencing, and the low frequency mutations. Next is the versatility. So the versatility. So NGS can be applied to a wide range of applications, including whole genome sequence, exome sequence, and total exome sequencing, which is called as the exome sequencing, and the transcriptome sequencing. So what about the key advantages of NGS for the degraded data? As I already mentioned, in the forensic scenario, we're getting the degraded DNA. The chance of getting the degraded DNA is much more higher in case of forensic science. So what about the advantages of the NGS for degraded DNA? The first one is the high sensitivity. So the NGS can generate a large number of short reads for even the small amount of DNA. If the amount of the DNA is very small, so NGS can generate a large number of short reads. This makes it ideal for analyzing the degraded DNA samples where the starting material may be limited. Next is the massively parallel sequencing. NGS can sequence millions of DNA fragments simultaneously, increasing the chance of obtaining sufficient coverage of the target region during the presence of degradation. So what about the target sequencing? So by focusing the specific region of the interest, NGS can maximize the yield of informative data from limited DNA sequence samples. So if we are getting the fragmented DNA or degraded DNA, because in that particular region of the DNA, so we are getting very less quantity of the DNA in that particular region of the DNA. So mainly if we are getting the very small amount of DNA samples, so NGS can maximize the yield of informative data from the limited DNA samples. This targeted approach can be particularly useful for degraded DNA where the overall quality may be compromised. So in targeted sequence, by focusing on the specific region of the interest, means we are not going for the focusing of the whole genome. We are only focusing on the specific region of interest. For example, my specific region of interest in the chromosome number 3, or we are focusing on the specific region of the interest of, for example, FTO gene. So if we are analyze, or if we want to know the genotyping of the FTO gene. So definitely there has been, it's helpful through the targeting sequencing. Then there is the error corrections. So the in error correction, advanced bioinformatics tools can be used to correct error introduced by DNA damage or sequencing error, improving the accuracy of the final results. So there are some kinds of specific NGS technique which is used for degraded DNA. What is something is the shotgun sequencing. So the shotgun sequencing is this approach involves randomly fragmenting the DNA and sequencing the resulting fragments. While it can be effective for high quality DNA, it may not be optimal for the degraded sample. Next is the target enrichment. So in target enrichment, this technique focuses on the specific region of the interest, such as the coding region and known genetic markers. And so we are using some STR markers to identify the individuals. So, through NGS, we are only targeting this specific region. So it will be helpful for profiling. It will be helpful for profiling DNA profiling from degraded DNA sample. Then single molecular real-time sequencing. So this technology can sequence long reads, which can be particularly useful for analyzing the highly fragmented DNA. However, it's more expensive than the NGS method. So as NGS has a potential to revolutionize the forensic investigation, leading to more accurate and timely identifications of individuals, reconstruction of crime scenes, and resolution of complex cases. So this is my references, and thank you.